So thank you for the opportunity to talk to you today about how we can learn from nature in order to preserve and improve the preservation of cell therapies. So there's two people on this slide, former President Jimmy Carter and Robin Roberts, who is a TV host on Good Morning America. So what do these two people have in common? Well, I don't know them personally, so I can't comment on the full nature of their relationship, but both of these people are alive today because of cell therapy. And they join the roughly 10,000 people a year who are treated with cells. So as the name implies, a cell therapy is a cell that's given to treat a disease or heal an injury. And on the left-hand side of this slide, we see some of the cell types that are being used to treat patients. And what's interesting is the other side of the slide, the right-hand slide, has a much longer list of diseases that are being treated with cells. So what that means is that certain cell types have the ability to treat more than one disease. The goal, the long-term goal, is that cell therapy be the fourth pillar of medical care. Most of us are familiar with pharmaceuticals or drugs, biologics, and the most common example of that is vaccines. Medical devices, Minnesota is a home to many medical device companies. So we want cell therapies to be the fourth pillar. Now, in order for that to happen, there has to be an infrastructure that permits that. There has to be methods to manufacture these cells. There has to be the ability to get approval from the Food and Drug Administration. Insurance companies need to reimburse the cost of those treatments. And finally, physicians need to actually prescribe cell therapies to their patients who have the appropriate disease. So preservation is a critical element of the infrastructure that goes into having the cell therapy available. There is a very, very unique supply chain when we talk about cell therapies. If we have a cell therapy, we have to harvest cells from a living human donor, and that typically takes place in a clinical setting, a doctor's office, a hospital, whatever. Now, you can't just process the cells there. The cells have to go to a special facility. At the University of Minnesota, that facility is on the St. Paul campus, and it is a special facility for processing cells. It may be ultimately infused into a patient, a completely different patient, at another location and potentially even another country. Now, because these are biological cells and the cells have to function, we have to keep the cells alive across the time and distance that it takes to go amongst these different sites. This is completely different than a pill. A pill might be produced in a factory, put into a bottle or container. You either get it from a pharmacist or from Target or some other store. So the supply chain for a drug for cell therapies looks nothing like the supply chain for a drug and represents a significant challenge. So I was sitting in my office looking out the window. It's a winter day, and I'm trying to figure out how to improve the preservation of cells used therapeutically. And I looked out at a tree that I can see from my window, and I said, well, how do trees survive Minnesota winters? That's an extreme environment, especially after this last winter. What they do is they express molecules that protect the cells against the cold. And those molecules are called osmolytes. But trees are not alone. Nature uses these molecules to stabilize a lot of biological systems. So on the left-hand side of the slide, the top two biological systems, the tardigrade and the resurrection plant, can survive desiccation, in other words, no water, for up to a decade. The bottom, the shrimp brine cysts, they can exist in very saline environments like the Great Salt Lake. And all of these systems use osmolytes to stabilize the cells. You and I use osmolytes to stabilize parts of our body. Our kidney is subjected to a lot of stresses with the toxins that we secrete in our urine. And as a result, our kidney expresses osmolytes to stabilize the cells and protect them. It's important to note that it's not just a single osmolyte, but actually a combination of osmolytes that are used by these biological systems to stabilize themselves. So what's an osmolyte? Well, we decided to choose, amongst all the different osmolytes that are out there, 3 different categories. We chose sugars, sugar alcohols, and amino acids. Most of us are familiar with sugar. In fact, some of you may have had it in your coffee this morning. So it is a sweetener. Sugar alcohols are also commonly used in food sweetening. And in fact, there are a lot of chewing gums that use sugar alcohols to sweeten them. And then amino acids are the building blocks for proteins. So our bodies produce amino acids, we eat amino acids. This is something, once again, found in nature. So we were bold. We took the hypothesis that we were going to imitate nature, and we were going to use a combination of osmolytes to preserve cells. The first cell type we were interested in looking at is mesenchymal stromal cells, MSCs. Everybody in this room has MSCs in their body. Okay? They're an adult stem cell. And they have a variety of biological functions, but a couple of the functions include improving wound healing and also modulating your immune system. Now, those properties, those important biological properties, are such that they can be used to treat a variety of diseases. And on the right-hand side of the slide, you see a listing of some of the diseases that are being used, they're being treated with MSCs. It includes cardiovascular diseases, it includes blood diseases, autoimmune diseases, and arthritis. So right now, there are over 200 clinical trials that are being pursued using these cells to treat all these different diseases. And in fact, over 3,000 patients have been treated with MSCs. And the state of Minnesota is where more patients are being treated with MSCs than any other state in the country, because there are active clinical trials both here and at the Mayo Clinic. Unfortunately, our ability to treat all the people who could benefit from MSCs is limited by poor methods of preservation. When the cells are thawed, their function post-thaw is not what it needs to be, and that has limited the number of patients that can be treated. So we went into the lab, we took our hypothesis, and we said, we determined that a combination of sucrose, glycerol, and isoleucine gave us the desired benefit. Now, one of the things we had to look at was how do the cells function post-thaw? And one of the problems with the existing methods is that the cells did not attach. And when they don't attach, they can't function properly. So the slide in the or the micrograph in the lower right-hand side shows a beautifully colored slide of cells, MSCs, post-thaw, that are attaching to a slide. And so we see not only do the cells attach, but when we stain in red the cytoskeleton, we see that the cell cytoskeleton exhibits properties that are much more like fresh cells than frozen cells. Now, we were interested in understanding what was going on inside the cells, so we looked at genes, and we looked at a lot of different genes. And I won't go into all of those details, but I will tell you that the cells expressed protective genes. When the cells are normally frozen, they express a lot of stress genes. And instead, when they're exposed to these osmolyte solutions, they express protective genes, the same kind of genes that our tree expresses when it prepares for winter. So these osmolytes are changing the behavior of the cell. Now, all puns intended, there is a sweet spot, okay? We're the only place in the country where we freeze cells down and we image them at -50 degrees Celsius, which is pretty cold. And based on these images, we can render individual images that tell us things about different molecules. So on the left-hand side, we have an image that's rendered on ice. So ice is bright colored, and places that are not ice is dark. In the middle, we see where the osmolytes are located. They can be in the areas where they're bright. On the areas where they're dark, there's no osmolytes. And then on the right-hand side, we see where the cell itself is located. And the cell itself is in the bright area, and the dark area is not where the cell is. Now, this allows us to understand at very high resolution how the cell is responding to this environment. And the distinct difference between the top row and the bottom row is that the cell in the bottom has a big chunk of ice in it. And you can see that. That cell's dead. That cell is not alive anymore. And what's even more interesting is the difference in composition between these two cells is very, very slight. Only slight differences in the concentration of sugar and slight differences in the concentration of amino acid. So you have to find that sweet spot. Now, we do have other good news, however, in that we have seen this behavior where the combination of these three molecules protects cells in other combinations of molecules. So it's not just one combination. So this is something, and that doesn't surprise us because nature expresses all kinds of different osmolytes in all kinds of different organisms. So what's the future for where we're going with this? We have these sugars, these sugar alcohols, and these amino acids. Well, we're currently working on being able to preserve other cell therapies. We're looking at preserving T cells and natural killer cells that are being used for the treatment of cancer. And in fact, these therapies are what saved President Carter from the metastatic melanoma that he was suffering from. We're also interested in moving our research into translation, into the clinic, so that people can actually benefit from our improved methods of preserving MSCs. But we're not done. And nature is not done giving us wisdom. In the left-hand corner is Rana sylvatica, common wood frog. And below that is the range of that frog and where it lives. It lives in principally northern climates. And in fact, the frog freezes in the winter. It freezes. It's frozen right there. In the spring, when it thaws, it's alive. So that frog uses 5 different strategies to survive the stresses of winter in those northern climates. One of those strategies are these osmolytes. We think that we can use some of those other strategies, the 4 other strategies, to help us preserve tissues that also have tremendous therapeutic potential. So we're not done learning from nature. So really what we're talking about here in a very fundamental way is completing this infrastructure for cell therapy so it can become the 4th pillar of healthcare. We do this by solving the very unique supply chain issues associated with this very unique form of therapy. And this should enable more patients to be treated by cells. And wouldn't it be nice if you could be treated in your doctor's office rather than having to come to the University of Minnesota to get your treatment? So effective methods of preservation will not only enable more patients to benefit, but also enable different contexts for that treatment, whether it's a doctor's office or a battlefield. Finally, I'd like to also thank the team that has worked with me on these problems, my faculty coordinators, faculty collaborators, Dave McKenna, Peter Dosa, Andre Van Wingen, and a group of very talented doctoral students, Qixing, Katie Pollack, Katie Hornberger, Roy Lee, and Guanling Yu, and of course, the financial support of the federal government. Thank you very much.